Around us, a vast, invisible world thrives, hidden just beyond the reach of our human eyes. But we have a window into this realm. The microscope is more than just a lens, it is an instrument of physics. By manipulating light, we reveal what the naked eye simply cannot see. Today, we explore the bridge between the physics of light and the discovery of life. Light is more than just what we see. It is an electromagnetic wave, a fundamental carrier of energy and information. Every light wave has a frequency, a rate of oscillation. In the world of microscopy, we perceive these different frequencies as different colors. These frequencies are the raw data of the microscopic world. By choosing specific light colors, we control our ability to resolve tiny structures, as shorter waves provide sharper detail. Resolution is the ability to distinguish two separate points in space. It is the fundamental measure of clarity in any imaging system. The human eye is limited. We can only distinguish objects down to about 0.1 millimeters. Anything smaller than that simply merges into a blur. Most microbes are significantly smaller than this threshold. Without advanced optics, they remain completely invisible, hidden by our own physical limitations. To bridge this gap, we use microscopy. By manipulating light, we can push beyond the limits of human vision to resolve the microscopic world. Brightfield microscopy is the most common form of light microscopy. In this setup, light from a source at the bottom travels directly up through the specimen. Because the light passes straight through, the internal structures of the specimen absorb some of that light. This results in a dark image or silhouette set against a bright background. Most biological cells are composed primarily of water, making them nearly invisible under a standard Brightfield microscope. Because light passes through these transparent structures with almost no absorption, they offer very little contrast against the bright white background. Traditionally, scientists have relied on chemical stains to add contrast. This dyes specific cellular structures, making them stand out against the background. However, there is a major downside. Most chemical stains require fixing the cell, which halts all life processes and effectively kills the specimen, preventing us from observing living, dynamic behavior. Darkfield microscopy overcomes the transparency issue by blocking direct light. By placing a stop in the condenser, only oblique light strikes the specimen. When this light scatters, it creates a high-contrast silhouette against a dark, empty field. Consider the bacterium Borrelia burgdorferi, the causative agent of Lyme disease. In a standard Brightfield microscope, this thin, translucent spirochete is almost impossible to detect because it doesn't absorb or scatter enough light. Darkfield microscopy changes the game. By blocking the direct light and capturing only the light scattered by the specimen, the bacteria appear as brilliant, glowing spirals against a deep, dark backdrop. This stark contrast allows researchers to clearly identify the pathogen, proving that with the right physics, even the most elusive structures can be brought into focus. The evolution of microscopy is a journey from simple glass lenses to sophisticated computational platforms. Yet beneath every advancement, the fundamental physics of refraction remains the constant foundation. Early observation relied on single lenses. As science progressed, these evolved into complex compound microscopes, allowing us to delve deeper into the cellular realm. Today, in 2026, we have transcended pure optics. Modern instruments now integrate digital sensors and artificial intelligence, processing light data in ways previously unimagined. No matter how complex the instrument, our core principles stay the same. We manipulate light's path to resolve the unseen, bridging the gap between simple glass and global discovery. Many biological samples are nearly transparent, making them invisible under a standard Brightfield microscope. To see them, we need contrast. Simple staining uses a single dye to color the entire organism. This makes the shape and size of the cells instantly visible against the bright background. Differential staining, like the famous Gram stain, uses multiple dyes to distinguish between different types of organisms or structures. One type takes on one color, while another takes on a different one. By adding these stains, we transform invisible life into high-contrast images, allowing scientists to identify and analyze the microbial world effectively. For centuries, microscopy relied solely on glass lenses to bend light. This analog process was limited by the physical properties of glass and the diffraction of light waves. By 2026, we have moved into the era of computational microscopy. We no longer just focus light. We capture raw data and use artificial intelligence to reconstruct images. AI algorithms can now decode the hidden information in light waves, effectively bypassing the old physical limitations. This allows us to see structures with clarity that were once considered invisible. Modern microscopy is no longer just about glass lenses. It is about computational intelligence. By training algorithms on millions of images, AI can predict and sharpen the blurry light data captured by the objective lens, essentially bypassing the diffraction limit of physics. Historically, observing live cells required staining, which often killed them. Today, phase contrast 2.0 allows us to image cells without these toxic chemicals. When light waves pass through the dense structures of a cell, they slow down. This creates a tiny phase shift, which is usually invisible to the human eye. Phase contrast 2.0 uses digital sensors to detect these precise changes in the light wave's phase, reconstructing the cell's structure with high clarity. This label-free imaging is a massive leap forward, keeping cells alive and healthy while revealing their internal functions in real time. Pathology has entered the digital age. Traditionally, pathologists examined physical glass slides under a microscope. Now, we use high-resolution scanners to create virtual slides. A virtual slide is a massive, high-fidelity digital image of the entire specimen. This allows pathologists to zoom into any region with extreme clarity. This technology enables remote diagnosis. A pathogen identified in a local clinic can be instantly reviewed by a specialist on the other side of the world. This is vital for identifying critical threats like Bacillus anthracis. Digital scans ensure that expert analysis is just a click away, drastically reducing the time needed for life-saving triage. In a modern clinic, every diagnosis begins with a sample. Triage is the first step in determining what needs urgent attention. Using Brightfield microscopy, clinicians identify standard cell types. It is the reliable foundation for sorting through clinical data. Today, digital tools amplify this process. AI systems instantly scan slides, flagging anomalies that might need a specialist's eye. This combination of classic physics and digital speed ensures that clinical decisions are faster and more accurate than ever before. To understand how bacteria react to antibiotics, scientists use a powerful tool called fluorescence lifetime imaging, or FLIM. Unlike standard imaging that just shows a static image, FLIM measures the precise time a molecule stays in an excited state after a pulse of light. When we introduce antibiotics, the chemical environment inside the bacteria changes, which alters this decay time. Researchers track these shifts in real time, watching as the bacterial response unfolds directly inside the tissue. This allows us to see not just if a cell is present, but how its metabolic health is changing in response to treatment. A traditional microscope shows us the what of a microbe, its shape and size. But in clinical diagnostics, knowing the shape isn't always enough to understand how a pathogen behaves. To see the why, the metabolic state, researchers now integrate Raman spectroscopy. This technique maps the molecular vibrations within the cell, revealing chemical signatures of life. By combining optical imaging with Raman data, we create a multimodal map. We don't just see the cell's outer shell, we see its internal chemistry, allowing us to distinguish active, dangerous microbes from dormant ones. Multimodal imaging is the new frontier in microbiology. It turns every visual observation into a rich, data-driven diagnostic, providing a complete picture of the microscopic world. In modern laboratories, the process of analyzing samples has been transformed by automation. Instead of a human scanning every square millimeter of a slide, machine learning algorithms now take the lead. Advanced digital scanners traverse thousands of fields of view. These systems process complex images in real time, effectively identifying patterns that might take a human researcher hours to locate. When the algorithm detects a cluster that matches known markers of disease, it instantly flags the location. This digital precision ensures that no potential threat is missed, even in a vast sea of data. By pre-screening the slide, automation allows human experts to focus their energy solely on these flagged areas. This synergy between AI-driven diagnostics and expert oversight is the future of clinical pathology. In the modern laboratory, digital tools like AI are revolutionary. But experts agree, AI is only as good as the physical data it receives. An AI algorithm can only interpret what the high-quality optics have captured first. Physics remains the king of the microscopic world. AI cannot create data that the optics failed to resolve. Physics remains the ultimate foundation of all microscopy. Today, microscopy is undergoing a fundamental shift. We are moving from simple visual observation to a world of data-driven science. Modern diagnostic platforms are increasingly cloud-integrated, allowing high-resolution imagery to be shared and analyzed globally in real time. We are seeing a 12% year-over-year increase in the adoption of these cloud-based diagnostic platforms. This evolution transforms every captured image into actionable data, fueling the next generation of automated discovery. We have looked deeper than ever before. Every discovery is a testament to our mastery over light. Centuries of scientific effort have bridged the gap between our eyes and the invisible structures of life. Today, we don't just observe, we understand. By controlling the smallest frequencies, we unlock the secrets of our biology. The world is waiting. Keep looking.